So hello, I will talk about the innate immune responses. I'll talk a lot about toll-like receptors and antigen-presenting cells and how you mount an immune response against a pathogen. I won't talk about complement because I'm assuming Michael Carroll talked about that already. I won't talk about CD8 T cells because I'm assuming that Michael Starnbach and Judy Lieberman have talked about these already. So I'll really focus on antigen-presenting cells. And it will be with regard to tropical diseases mostly. So as I normally do even for all my classes, it's the usual thing. You know, you're bombarded by all these pathogens all the time. You're on the bus, you're on the train, you're at home with your kids, they're coughing, hacking all over you, and it's why aren't you dead yet? And it's because, as you've already heard, your body has evolved over time, has certain innate pre-existing immune responses which are able to recognize anything from viruses up to, as I'll talk about today, worms. And they can recognize these and cope with them enough to where you can live and survive this acute infection until, of course, the adaptive immune response, which you all know about as having antibodies made and effector T cells. And this is all done by specialized cell types, these antigen-presenting cells, macrophages and dendritic cells, that have these receptors on their surfaces which allow them to recognize patterns of what are called commonly displayed antigens. So like bacteria that are gram-negative have a specialized pattern that groups of them have. Gram-positive have others. Yeast have another type of a sugar on them. Tuberculosis has another type, and worms have their own. So they're families of commonly shared molecules. And you've already heard you've got intrinsic epithelial barriers to infection. So obviously your skin prevents you from infection. You know what happens when you run into something in the backyard or a rusty nail. You get a site of inflammation or redness there. Those are all your immune effector cells coming into the site where pathogens might be. And in fact, you could break it down as to these are the natural barriers you have: skin, gut, lungs, eyes, nose. These four being mucosal responses, and they're mechanical. Okay, you've got, of course, across your skin epithelial cells that form very tight barriers against pathogens getting in. In the lungs, you have this flow of air or fluid, also in the gut, and movement of mucus bacilli. And in humans, of course, the mucus layer is extremely thick, which is good and bad. Chemical, there are lots of different things that are on the skin, as well as your own inherent bacterial flora that you have, that you need, that help prevent other things from coming in. And you actually make antibacterial peptides, and of course in the salivary glands you make lysozyme, which breaks down bacterial cell walls. So as I mentioned really early, your immune system has to cope with this unbelievable array of pathogens, right? From things as small as viruses to items as large as worms, tapeworms being as long as 20 meters in the human body. In the developing world, you see a lot of people infected with worms, Ascaris, roundworm nematodes that are as long as a pencil. And in fact, where we were in Brazil, there are sometimes when they treat the kids, they'll pass a mayonnaise jar worth of these Ascaris parasites out. So your body, of course, you have to be able to cope with all of these things, and it does. Now, obviously, while a virus and bacteria can be seen by an antigen-presenting cell or a macrophage, and they can bind to them and phagocytose them and then kill them intracellularly, something as large as a pencil or longer is not a phagocytosable surface, okay? So you have to have a way to cope with these organisms as well. So you have to cope with infected cells, i.e. when viruses infect you. You have to cope with single-celled extracellular pathogens, bacteria, protozoans like malaria, trypanosomes, and of course the big guys, the helminths. And there are, of course, pathogens which invade the mucosa, as well as those that are luminal. And please, I guess we can stop for questions at any time. So you have these different pathogens classes that I've already talked about: extracellular bacteria, parasites and fungi, intracellular bacteria and parasites, TB, leishmania, and malaria, viruses, intracellular, and of course these parasitic worms, which are always extracellular, those that reside in the blood system or they reside in the gut. So you've heard a little bit already from Michael Carroll and probably Michael Starnbach on this innate versus adaptive, and I've sort of already alluded to it. So as I mentioned, innate responses have evolved. They're pre-existing These cells in your body directly recognize invading pathogens via these patterns of arrays on their surfaces. These are antigen-presenting cells, neutrophils, NK cells, and another class of antibody-forming cell called B1 cells. B1 cells are a very primitive form of antibody-producing cell. They're found in the lungs, and they're found in the peritoneal cavity. So if you were to think about what would be in the peritoneal cavity, why would you want to have cells in the peritoneal cavity? Okay. So let's think, if you're a primitive caveman, what's your diet? Whatever you could find, probably along with a little sticks, stones, and bones, right? Which might have done what into your intestinal tract? Caused perforation. So what you really then, these had to evolve then to cope with the fact that primitive man didn't eat T-bone steak or tofu or whatever, but he had to deal with whatever he could get, and therefore they got numerous perforations. So in fact, when you look at the repertoire of antibodies that these B1 cells make, or these neutrophils, they're all antibacterial cell surfaces, very conserved things that primitive man would have had in his gut. Same thing would occur in the lungs, right? You have lots of chances for injury. Okay, now on the other hand, adaptive responses, these form in response to a pathogen, but they're delayed, and I'll show you a little bit about this. So the T cells, both CD4 and CD8s, mature following the presentation of these pathogen antigens by the innate cells or the antigen presenting cells. B cells similarly will react with these guys as well as T helper cells, and they can also interact with T cells to help them mature. So T cells, these effector T cells that you hear about so often, whether they're CD4 T helper cells, or as Michael Starnbach and Judy Lieberman talked about, CD8 effector cytotoxic killer cells, they respond after being educated by antigen presenting cells. They're antigen specific. So I'm going to talk a lot today about these two classes of antigen presenting cells, macrophages and dendritic cells. And what they're good at is they're good at surveying your body. Okay, so you have these under your skin. You have them in all your mucosal sites. Okay, and if you think about it, where are you going to encounter a pathogen? Right, you're either going to ingest it, you're going to breathe it, or it's going to break your skin barrier. So this is where you want these antigen or pathogen sampling cells, are in these sites. And they're very good with their receptors at sampling the environment. So they know immediately if a foreign particle or pathogens come into the area. So obviously we could go bonkers here, but for me, this innate immune response comes into three parts. The first part, which I've just sort of mentioned to you, is this recognition phase. So you've got these antigen presenting cells, whether they're macrophages or dendritic cells here or in your lungs or mucosa, that recognize a pathogen. This recognition leads to them becoming activated, and then they then mature into functional cells that will interact with your CD4 T helper cells and CD8 T helper cells, present pathogen antigens, and now activate the adaptive immune response. So, as I already mentioned to you, these immature dendritic cells reside in the peripheral tissues, and they actually do look like that. That's why these are called dendrites coming off of them. So when you look at them, they're not like a round mononuclear cell. They actually have these processes coming out, and that's what they're using to probe the environment. They're very sticky, and if they encounter a pathogen, okay, they engulf it. They then will migrate via the lymphatic system to the regional lymph nodes, and in this case, they'll now present the antigens to your CD4 and CD8 T cells and get the adaptive immune response going. So the innate immune response is actually required to clear bacterial infections. So what I'm going to show you are just a couple of cartoon figures from experiments done that show that bacterial infection in mice that are deficient of these innate cells, these neutrophils or macrophages, can't cope with the infection. So you compare them to mice which have neutrophils and macrophages but no T cells or B cells. And the same could be seen in humans that have immune deficiency diseases. So here's your level of bacteria, okay? Here's your duration of infection. And in humans or mice that lack these effector innate cells, macrophages or polymorphonuclear cells or neutrophils, you can't control it. Bacteria go off the wall and you croak. in these SCIDs, severe combined immune deficiency mice, they have no functional T or B cells. Or in RAGs, they can cope, okay, so they do better. So they have functional innate cells, so they control the initial wave of bacteria, right? But they can't keep it down because they can't form an adaptive response. And of course, in normal intact animals that have both the innate cells as well as the T cells and B cells, then they control the infection, as we all know. So you require both. So I've talked a lot about this pattern recognition. Okay, so then how do these cells recognize pathogens? So they have receptors that were cleverly called pattern recognition receptors, and they do recognize these conserved patterns of bacterial or viral antigens. And Michael Carroll talked to you about one group of these, the complement receptors. So pathogen-associated molecular patterns. So opposite of these pattern recognition receptors then must be these conserved patterns of antigens or molecules on the surfaces of these bacteria, viruses, worms, etc. Okay, they're called pathogen-associated molecular patterns, or some people call them PAMPs. The best studied of these are, of course, from bacteria. They're almost exclusively, and I'll show you a cartoon in a minute, molecules that are found in the bacterial cell walls or their membranes that are unique to these classes. So like I told you before, there are gram-negative and gram-positive bacteria, and they have different types of classes of these molecules on their surface. So I don't know how much you're aware of this, but let's say that an example would be lipopolysaccharide or endotoxin. And a lot of you may be aware of this sepsis. So if you get an infection and they can't get it controlled, you begin having spiking fevers and chills. This is sepsis. It's a response to endotoxin. People die from toxic shock or sepsis. So if you look here, in a gram-positive bacteria, you have these types of molecules on the surface, and these are just for examples. Okay, and these are all each different classes of these PAMPs. For gram-negative bacteria, you have the more well-known one, this endotoxin, this lipopolysaccharide. Okay, and they also vary by species. So these innate receptors, so these surface membranes, so I've told you these dendritic cells are there working their way around your body, through your skin, through your mucosal membranes. And they've got these receptors on their surfaces. They're covered with these pattern recognition receptors. Okay, some of them are complement receptors. Some of them are what are called lectins. They see specific types of sugars like the macrophage mannose receptor. These are all evolutionarily primitive, okay? They bind these patterns of bacterial sugars and lipids. And why I say that they're evolutionarily primitive, if you go back into insects like Drosophila, the fruit fly, they express many of these same types of receptors on their cells, even down to sponges. In fact, the toll-like receptors that I'm going to talk to you about today were first discovered in fruit flies. And they were thought to be a developmental marker, and it turns out that they actually form part of the innate immune response in insects as well. So what are these? Well, right now, there are at least 11 different toll-like receptors that have been described, and I'm sure that there will probably be some more coming up. Not all of these, and we call them TLRs, toll-like receptors, not all toll-like receptors directly bind the PAMP, or the pathogen-associated molecule. Okay, so then you're going to say, well, then how do they work? Often what happens is the PAMP binds another molecule on the surface of the antigen-presenting cell, which then traffics it to the toll-like receptor and therefore engages and activates the cell. And I'll show you a cartoon of this, I hope, if the movie works, in a minute. So PAMPs from bacteria generally signal through these toll-like receptors and activate your antigen-presenting cells, alerting your body and them that you are now infected. And again, the one that's most commonly studied is bacterial endotoxin, and it's recognized by something called toll-like receptor 4. So if you were just to look at a cartoon of your effector antigen-presenting cells, they have this mannose receptor that sees sugars that are found on a lot of pathogens. Lipopolysaccharide receptor. Here's toll-like receptor 4, another type of glucan receptor found on yeast, and other scavenger receptors. And when these bacterial pathogens bind, okay, They cause these receptors to come together on the cell, and I'll show you this cartoon, which leads to activation of the cell. Remember I told you that there was recognition, activation, and maturation. So they recognize, they become activated through a signaling process, and this leads them to their maturation, which allows them now to traffic, remember, via the lymphatics to the lymph nodes, where they'll now present the pathogen molecules to your T cells to begin this adaptive immune response. And part of this, we won't talk a lot about it, but you might have heard it from others, part of this involves the production by these cells after activation of cytokines and chemokines. So these are soluble factors which are involved in maturation not only of the antigen-presenting cells, but also of the T cells. Okay, so let's skip this. So, not that I want to scare you to death, but remember I told you about not all pathogens or PAMPs bind directly to TLRs, toll-like receptors. So this is done by a group at Oxford University called the Triantafyllos, two sisters, two PhDs that work together. They study innate immune responses, specifically with toll-like receptors. And what they found is if they do experiments where they, do you guys understand, do you know what about immunoprecipitations? You know, so if I add, okay, so if I had an antibody to this molecule here, and I added it to these cells, it would bind to that, but it could also bring down, in an immunoprecipitation, I could bring down any adjacent molecules associated with it. So by doing these experiments, they found out that it's not just this molecule, but all of these other molecules are associated with endotoxin, LPS binding here. And in fact, LPS doesn't bind here. It binds over here, here, and with all of these, which then come together in a complex, activating the cells. So we're going to talk a little bit about these dendritic cells. So dendritic cells in your body are the most specialized and the most active of all your antigen-presenting cells. They're spectacular at surveying your body for pathogens. And the association of this bacterial endotoxin with TLR4 activates these signaling events, okay, which occurs through adapter proteins. And we won't get into this in big detail, but one of them is called MyD88. This eventually leads to the activation of transcription factors, right? The cells then produce these cytokines and chemokines because the transcription factors are there, which then now leads to their maturation and also then affects the subsequent T cells they interact with in the lymph nodes. So it's really critical. And this is another cartoon for you. So here's this mammalian toll-like receptor pathway. So here's a toll-like receptor X. It has this extracellular domain. So here's if your cell membrane of your antigen-presenting cell, and it's sticking out here, it gets activated, right? It binds, and it sends a signal through here through MyD88, this adapter molecule, which then goes through this whole complex thing. And hopefully, again, like I said, I'll show you this cartoon movie of how it works to where this molecule here called NF-kappa B goes into the nucleus and starts the synthesis of genes for cytokines and chemokines. So here's another cartoon, again being a little redundant. So this endotoxin binds to this molecule on the surface. It's not TLR4, LPS binding protein. That then associates with TLR4, causing activation and release of these factors so that NF-kappa B can get into the nucleus and start transcription. Okay. So as I said, once this signaling activates the transcription factor NF-kappa B, it translocates to the nucleus, which in turn turns on the production of these pro-inflammatory mediators. So when I say pro-inflammatory, what normally happens to you guys when you get a wound? It swells up, right? Okay, so you get a pro-inflammatory, or when you get a viral infection, you get a fever. Okay, so you get a pro-inflammatory response. So these cells are making these pro-inflammatory mediators, these cytokines, these chemokines, prominently IL-12, TNF-alpha, and IL-6. Now IL-12 is key, and some of you that were following early anti-cancer trials that were done at the Brigham back in the 90s might remember that this was the wonder drug. It's the cytokine that was going to cure all cancers. It's so potent that when they injected it into the 6 volunteers, it killed them all immediately. Okay, so they died from a pro-inflammatory response due to the potency of this particular molecule. So it's, as you can imagine, it's been dropped as a cancer treatment. So what is the role of this adaptor protein I talked to you about? So downstream of these toll-like receptors are these adaptor proteins that initiate these cell signalings and activation. I'm going to show you, and you'll see it in this cartoon, the default pathway for almost all toll-like receptors appears to be this molecule people call MyD88. Okay, so this, if you looked at the response to endotoxin or to Mannan or to any of these other bacterial molecules that activate your antigen-presenting cells or dendritic cells through toll-like receptors, it appears to always go through this molecule MyD88. So what happens then if I take mice and we knock out the genes for MyD88? So now they're deficient. They don't have this pathway that will allow you to activate your antigen-presenting cells properly. So we're going to show you some experiments where I actually do this in mice that are then infected with this pathogen called Toxoplasma gondii. And most of you familiar with toxoplasmosis? Okay, so it's a protozoan parasite. Okay, and we've already showed you this, but this is just to reiterate. Here's your tolls, whether it's toll 4 or toll 9, MyD88 appears to be the key adapter molecule. So when they refer to wild type mice, they mean normal, genetically intact mice, okay? Or MyD88 null mice, those that have had the gene knocked out. So if you infect them intraperitoneally with Toxoplasma gondii in a wild type mouse or this mouse, here's what happens. If you look at the levels of cytokines, either IL-12 or interferon gamma, that have been associated with the ability to clear this infection, what you find is that in the wild type mouse, you make very nice prodigious levels of these cytokines, both. But in the mouse that we've now knocked out this adapter protein, they can't make these pro-inflammatory mediators. So in fact, it is critical for the ability of your cells to make these pro-inflammatory mediators. Okay, well, what does that mean? Well, what it means is that the wild type mice that make the IL-12 and the interferon gamma, they all survive. But the MyD88 knockout mice all die within 15 to 16 days post-infection because they cannot control the infection. So what do you think in this room? How many people do you think are infected with Toxoplasma? Percentage-wise? 10%? 20? 30? 20? Okay, probably somewhere in the range of 30 to 40%. Okay, so again, so why aren't you dead? Okay, so you have developed this nice immune response, right? You've controlled it. And in fact, maybe many of you know that this parasite resides in a latent form in our body. So what happens when you're immunocompromised? It comes up. So one of the things that people with AIDS have is they have neural toxoplasmosis. It's a serious problem. Okay, now let's see if we can do this. And I might need the lights down, but let's see. So I'm going to try to show you a movie done by a group at the University of Georgia. They're even a little more bonkers than I am. I know that's hard to believe, but they're called the Complex Carbohydrate Research Center. So whereas I'm interested in how these pathogen-associated carbohydrates activate our cells, they're interested in the specific molecules on the cell surfaces. So they went ahead and produced a movie, this one. And what they're going to show you, and I'll start over here. So instead of using a bacteria, and of course this is a cartoon, right, so things are exaggerated. So here's your cells. This is supposed to be a bacterial membrane array of this endotoxin. So to try to sum it up for you, what's going to happen, they're going to show this coming across these cell surfaces. They're going to show it binding to this first LPS binding molecule, CD14, coming together, then beginning to cross-link a toll-like receptor, then going into the cell and showing you all those complex signaling pathways I showed you, and eventually to NF-kappa B activation. That's if I can, this will play. So is that big enough for you guys? Okay, so here comes our larger than life bacterial membrane-associated endotoxin that you would have in the environment of an infection. And it's important to note that, remember, these are patterns, so it's not one molecule, they're large arrays of these molecules. So here it's coming across your cell surfaces. This is CD14. So CD14 has recognized the carbohydrates here on this bacterial endotoxin. Now bound it, begun some cross-linking. CD14, remember I told you about bringing stuff to toll-like receptors, is now going to bring this to toll-like receptors which bind another part of the molecule. They dimerize, so two come together, forming this activation complex. So here's your lipid bilayer. You can see this activation here coming through. So this is normally phosphorylation. Things become phosphorylated. Here's our good friend that we've talked about, MyD88. We won't talk about these, but these are other associated signaling molecules that are involved in the pathway before you get to NF-kappa B. Here's your nucleus with nuclear pores. And what's critical are this complex of molecules here. These molecules, the I-kappas and I-kappa beta, inhibit NF-kappa B from going to the nucleus. So they inhibit normal activation. But when you get this signaling complex, it breaks it apart, allowing NF-kappa B to go into the nuclear pore, and as we said, bind to transcriptional sites and start transcription of these cytokines and chemokines. And did I get that to you guys last time or not? I can't remember if I got the move. So I'll try it again. I'll call the Georgia people and see if they'll release it for you guys, and you could use it in your classes if you want to. Okay, so you've seen this activation phase, right? So now there's a maturation phase you have to have. We'll talk about that a little bit. So remember I told you that you make these things like IL-12 and interferon gamma and TNF. They're all pro-inflammatory. So you get the production of these cytokines and chemokines that now induces these activated dendritic cells and macrophages to mature. And as they begin doing this, they upregulate expression of other molecules on their surfaces, and those molecules are key to activating T cells in the lymph nodes. So if they don't upregulate these, and they're called B7-1, B7-2 in the major histocompatibility complex that presents antigens, then you don't get an effective immune response. So these mature dendritic cells migrate to the lymph nodes. They have activated co-stimulatory molecules, MHC class I. They present your pathogen antigens to naive T cells. Okay. And in the presence of these co-stimulatory molecules and the cytokines, they tell your T cells how to respond. Okay, so remember we talked about pro-inflammatory responses? You also have an anti-inflammatory response. So not everything is pro-inflammatory. So if we looked at this cartoon again, depicting a skin wound, so here's your skin, bacteria, you've been out in the garden or something and got whacked. Here come the bacteria. Here's your dendritic cell called a Langerhans cell in the skin, binding it, okay, activating it, going to the lymph nodes again. As it's migrating through, it's beginning to mature, upregulating these co-stimulatory molecules on its surface, as well as MHC class I and II, which are the molecules that will present bacterial antigens to the T cells, allowing you to then activate these T cells. So the innate response, the critical thing here is that the innate response that you make right off the bat will direct the subsequent adaptive response. All right, so how you respond initially with your dendritic cells dictates if you are alive or dead later, to be blunt, okay? So these dendritic cells, again, produce these cytokines and chemokines, and the types of cytokines produced direct the maturation of these CD4 T cells, commonly called T helper cells, to what's called a T helper 1 or a Th2 type. The Th1 is the response you're most often familiar with. It's pro-inflammatory. So when you get the bacterial infections, the viral infections, this is the type of response you get, almost default. Th2 type is anti-inflammatory, and you very rarely see this. But where you do see it is in people that are infected with parasitic worms, okay? So viruses, bacteria, and most protozoans drive pro-inflammatory responses, but worms, completely opposite, drive anti-inflammatory responses. Okay, so if this was, so let's say that this was the level of co-stimulatory molecules on the surface of my resting cell. When it's activated, this is the level. Okay, so I really mean that you've increased the number and their activation level. And the way that we would do that in the laboratory is we would stain with fluorescently labeled antibodies to those markers, and we'd run them on something called a fluorescence activated cell sorter, and it tells us the level of expression on each cell.
The anti-inflammatory drugs, there are plenty of them on the market.
Yes, there are.
Do they have a direct effect on the way in which these cells get rid of them?
Well, as you're probably aware, anti-inflammatory drugs have a lot of effects, not all of them good. So like for most people that have serious inflammatory diseases, let's take for example inflammatory bowel disease, colitis or Crohn's, you'd probably be on a corticosteroid like prednisone. Okay, prednisone is very effective at knocking down the inflammatory response. The only problem is prednisone has lots of terrible side effects and that you're now more susceptible to other illnesses, plus the accelerated appetite, water weight gain, all kinds of other problems. Prednisone is still used a lot. There are new drugs out now that are actually, the most potent one is actually an anti-receptor antibody. So it sees the TNF alpha receptor on these cells and blocks it. So you dampen it down. What does that do? Well, it works, but if you dampen down TNF alpha, which is a critical thing for your ability to respond to most infections and pathogens, then of course now you become more susceptible to other infections. So they do work, but of course all of them have some side effects. Anything else? Okay. So again, to be a little redundant, viruses and most bacteria, and I say most, induce this one we talked about already that was so important, IL-12, it's pro-inflammatory molecule, by dendritic cells. This can activate another type of cell we're not going to talk about here called natural killer cells, which then produce interferon gamma. This will also then direct Th1 pro-inflammatory type T helper responses. Other pathogens, and what they really mean here is what I've told you, worms, they don't activate dendritic cells to produce any IL-12, which is very bizarre. In fact, they cause the synthesis and secretion of a cytokine called IL-4, which drives anti-inflammatory responses and Th2 type responses. And IL-4 is also associated with something else you're probably aware of, allergy. You need IL-4 to make this molecule called IgE. It's an antibody associated with allergy. So to make a long story short here, hopefully, the whole story I've shown you here is how bacteria and viruses activate dendritic cells in a pro-inflammatory way. Those are called, like Th1s, those are called DC1s. And everybody understands that, and a lot of research has been done. In contrast, obviously we do mount anti-inflammatory immune responses, so one would think that there must be some way to activate cells to go the opposite way towards DC2s. However, much of the immunology community considers that this is not an active process, it's the default pathway. That in the absence of very strong stimulation by bacterial or virus PAMPs that would then drive this pro-inflammatory response, these DCs, as they become activated, mature by default to DC2s. So our group got interested in this whole idea, well, are there molecules on pathogens that drive these Th2 type anti-inflammatory responses? And again, to make a long story short, I've sort of already told you what it is. Parasitic worms drive anti-inflammatory responses. In fact, parasitic worms could shut down the response to bacteria. Parasitic worms in our field sites in Brazil and China, if I go in with a common childhood vaccine tetanus toxin, and I compare the vaccine take in kids who have worms versus those that don't, 50% fewer of the children who have worms convert a response to tetanus toxin. So these worms produce molecules that suppress your normal pro-inflammatory response, okay, even to vaccine antigens. Okay, so the worm that we normally work on in my lab and have for a long time is this worm called a schistosome. So it's a trematode parasite, a platyhelminth. It's a platyhelminth like the tapeworms, the flat segmented worms, except for these are normally, they're leaf shaped. And normally they have separate male and female they're hermaphroditic. They have male and females together. This particular one is the only one that has separate male and female sexes, okay? And this guy with the knobs on him, that's the male, and the female lives inside this canal here. And they move up and down your blood vessels off the mesenteric veins or off of your bladder, depending on your blood glucose and PCO2 levels. They actually migrate up and down using this sucker. And then they lay eggs, and the eggs are what causes the pathology and the disease. Okay. So this is where we work. And after teaching high school biology for a while, and then going back to school, being trained as a molecular biologist, an immunologist, I hadn't seen a lot of this stuff. So when I first went to Brazil, it looked like something right out of the 1940s tropical medicine textbooks. Nothing had changed. And in fact, these people are all working in water. They're washing their clothes. They're washing their dishes that are all contains the intermediate host, the snail. And what happens in this particular disease is when you're in the water, and let's say you defecate or urinate in the water, and you have eggs in your urine or feces, they hatch and they infect these snails. All this asexual multiplication goes on in the snails, and it's very nice the way this parasite evolved. So as the day gets warmer and hotter, these are photoperiodic organisms, they begin to emerge from the snails. And all they do is they swim up and down in the water. They don't care who they encounter, but whoever they encounter, they try to penetrate. And of course, the right host is humans. So when they penetrate you, they burrow right into your skin in about less than 1 minute, and you're infected. And again, the pathology occurs because, like most parasites, right, you've got to keep transmission going. You've got to find your intermediate host or your vector. So for malaria, mosquitoes bite you, they take up the infected red blood cells, etc. In this case, they've got to get the eggs out of your intestines into the water. So that's in the feces. But because you've got the blood flow normally going back to the portal, to the liver, about half the eggs get flushed into the liver where they're not supposed to be. It's a foreign object. You form a granuloma around the eggs to protect yourself, but in the meanwhile, you start destroying your liver tissue. This doesn't happen much anymore with the advent of very good drugs against the parasite. Okay, if you treat people frequently enough, they don't normally develop fibrosis. And what the fibrosis does is lead to something called portal hypertension. So for the last people that died in our village, they're 65 to 70. And it's because they develop portal hypertension, so they develop anastomosis. So the blood vessels can't get to the liver, right, because you've got so much back pressure. So they begin developing blood vessels which bypass the liver, your filtering organ, and they go right into your esophagus. So you get esophageal varices, they rupture, and you bleed to death. But anyway, back to dendritic cells. My digression, okay. So we've showed you that this is what can happen, right, to this point. I haven't shown you any of this yet, but I've shown you that you can take a naive dendritic cell, hit it with bacterial endotoxin, you get this activated dendritic cell that upregulates all of these molecules on the surface. And we talked about IL-12 and all these molecules leading to this pro-inflammatory T cell. No one knows what's going on here, yet we know we get these types of cells. The question is, how do you get there? So as we began looking at these parasites, we realized that these eggs of the parasite were the ones that drive these Th2 responses. And it doesn't matter if it's a schistosome or a filariid or a tapeworm or whatever, all of them have eggs or other stages that drive Th2 responses. And it turns out that last year, 2 other labs published papers supporting our original observations and other parasites showing that carbohydrates are exclusively responsible for driving these Th2 anti-inflammatory responses. In fact, you guys are familiar with C. elegans? Even C. elegans has the exact same sugar, so they're biologically conserved all the way down to C. elegans. So our goal then was, having found out this property, that if you remove the sugars, you can't drive Th2 responses, then could you possibly identify these sugars? And because I was crazy during my early years and made 5 zillion monoclonal antibodies, we were able to identify these. And in fact, for a long time we thought we weren't, my student and I, and I'll try to wrap this up quickly for you guys. we made these monoclonal antibodies to sugars, and the question is, how are you going to determine what they are? People work with proteins and peptides, right? Not with sugars. So we were at an MD PhD retreat, and this neurobiologist said, why aren't you screening these against day 9 or 10 mouse fetal brain tissue? And I'm like, why would I? And she says, well, because it's the richest source of variant glycoform structures known, because that's how your neural cells home, by the basis of sugars and sugar-lectin interactions. So I gave her the monoclonal antibodies. They lit up the mouse brain, and I thought that was a cool picture. It meant nothing to me. She says, well, your sugar is LNFP3, state-specific embryonic antigen 1. You can buy it from Sigma. So I'm talking to you now about a worm sugar, right? But in fact, it's a developmentally regulated sugar in humans and most mammals. Okay? And I don't know how much you guys know about sugars, probably not a lot. And there's no reason why you should. This is something called Lewis X. Okay, so Lewis X is a very well-known biologically interesting molecule. So where do you find these molecules in normal tissues? Well, you find them on, as I told you, developing fetal tissues. They disappear, and you find them again on a state where your cells become fetal-like or embryonic-like. Does anybody have an idea what that is? Cancer. Okay. So cancer cells express what's called the fetal embryonic antigens, and they re-express these sugars. So it appears now that parasites from viruses, we've now shown the sugar on HIV, viral envelope, from viruses up to worms, certain pathogens have now exploited this molecule to suppress the host immune system to their advantage. So I'll skip a lot of this and go to the punchline for you, but what we did is we got working with a company called Neos Technologies. They took our sugar molecule, they put a linker on it and put it on a carrier molecule so that we now mimic like a bacteria, like that cartoon I showed you. So we now have a molecule that can cross-link receptors on antigen-presenting cells. Let's go. And what it does is, here's our good friend LPS or endotoxin, whereas LPS drives high levels of bacterial-induced interferon gamma, right, Th1 pro-inflammatory, the sugar actually suppresses it. And in contrast, it drives, in fact, the Th2 or anti-inflammatory responses by upregulating the cytokine IL-4 that we showed you is associated with Th2. So in fact, it's able to suppress pro-inflammatory while driving this Th2. Okay, let's not do this. Okay, so to wrap it all up and go to something that might be interesting for you guys, what we've shown is that this sugar actually signals through the same toll receptor that bacteria do. Okay, so you're saying, well, that guy's really a nutcase, how can that possibly happen? Remember I told you there's a MIghty88 response and there's a MIghty88 independent response. It turns out that LPS, as I've showed you, signals through TLR4 MIghty88 dependent, and the sugar drives a MIghty88 independent pathway. And of course, that's where a lot of us are now working, is trying to figure out how does this happen. And remember that cartoon where I showed you all those different receptors that bind LPS that then associate with TLR4? So it's got to be then upstream here. It's got to be the molecules that bind to the sugar that then interact with TLR4 that then dictate it's MIghty88 independent and not dependent. And if you could figure all that out, you now have a potent class of anti-inflammatory molecules that could be used to treat autoimmune diseases, et cetera. And in fact, if you look at people in developing countries, what don't they have? They don't have autoimmunity. They don't have Crohn's. They don't have colitis. They don't have myasthenia gravis. They don't have muscular or multiple sclerosis. Any of these things. They don't have psoriasis. Okay, so in fact, what happens to them when they get to the developed countries? They begin developing autoimmune diseases. What's been happening in this country and in Western Europe for the last 20 years in terms of autoimmune diseases? They're going through the roof. They're going through the roof. Now, and some of you guys may disagree with me, and that's okay. But I'm a big proponent of dirt. It turns out as the cleaner we've gotten in our environment, then we've done less to educate our immune systems. And early education of the immune systems, I believe, is critical. And so I know there are a lot of moms out there that want to wipe their baby's hands down at every case, but it's probably not a good idea. I mean, clearly if you have meat on the counter or something like that, yeah, wipe them down. But if they've been outside playing, probably not a great idea. It's probably good to be exposed to many different things early on and educate your immune system. Let me close with this one last bit to show you that they work. Okay. So I've already told you that this sugar is a common molecule shared by our own cells, cancer and embryonic, as well as pathogens, and they drive these Th2 type responses and immune suppression. To further that, this is the only sugar commonly found in all races of humans in human milk, so it's biologically conserved. Human milk is immunosuppressive. Why would you want human milk to be immunosuppressive? Because the bacteria is taking in the baby's taking in bacteria for the first time. Bacteria have endotoxin that drives pro-inflammatory. You want to not have the baby have gastritis and colitis. It's found in buckets in the urine of pregnant women. Pregnant women are immunosuppressed. I've already told you it's found on cancer tumor cell types. It's found on the HIV-1 viral envelope. And for those of you, unfortunately, that have ulcers, Lewis X negative H. pylori drives Th1 responses, and Lewis X positive bacteria drive Th2 responses. So it is, in fact, found throughout biology. What does this mean in terms of autoimmune diseases? So this guy, Weinstock, Joel Weinstock, is a gastroenterologist in Iowa. He was watching a lot of our work, and I don't know what he was thinking, but he thought, well, instead of getting the sugars that Don Harn has, I'm just going to give my patients parasites. I'm going to give them worms. So what he did is he went out and he said, I'm not going to give them a human parasite or a worm, I'm going to give them a pig worm. So he got a pig nematode, you got the eggs, and the way you normally infect is you take it, the eggs hatch, they burrow into your mucosa. So he infects them with these, and he reports it in the most prestigious journal in the country, the New York Times, and claims that in fact all of his patients went into remission. Now, this is not the way I think the FDA would like to see an anti-inflammatory drug. However, he's now in phase 2 trials. And I was just called 2 weeks ago by a fellow at the Rockefeller who has Crohn's disease and saw our work. And the guy's pretty bright. He's a Harvard MD, PhD, who's now a professor there. He went all the way to Thailand. He took this bit of work, went to Thailand, got the human counterpart and infected himself with it, and said he's actually in remission. He realizes it's dangerous, but he's in remission. And he wants the sugars. Well, the sugars are, the company owns the sugars. I really can't go around giving the sugars out, but we are heading towards using this, and I'll show you why. So we tested our sugar conjugates, the ones that activate these dendritic cells, whether or not they would actually treat these things. So I'm going to show you 2 diseases that we worked on. One is psoriasis, and the other one is inflammatory bowel disease. And this was picked for us by the company because the company wants to make money, right? Psoriasis, 2% of all Caucasians have psoriasis, so it's a big market. And this mouse here, they spontaneously developed psoriasis beginning at 4 to 5 weeks of age. So what I'm going to show you is that we gave them 2 injections of the sugar or the control at weeks 4 and 6, stopped everything. 8 to 10 weeks of age, the mice were examined for psoriatic lesions. So I'll just show you a couple quick photos. So does this show up? So you can see these mice have developed this psoriasis-like lesions on their back. So these are the guys who got the controls, and these are the mice that got the sugar. In fact, if you look close, this guy's got a little bit of a lesion developing here. But remember, we weren't constantly treating them. We only gave them 2 shots and stopped. So in fact, this will actually stop, and we've now done experiments where we can actually treat. So they can develop psoriasis like the other mice, give them the sugar, and they'll get better. The other one's colitis. We won't worry about how it works, but it's another mouse model. They develop disease very early on, within 12 days. And people do this by looking at weight loss, diarrhea, prolapse rectum, et cetera. We're going to show you some pathology of the colon, because what normally happens is when people have colitis, it's really bad and flares up. They have to resect the colon periodically to where eventually they do a colostomy on somebody. So it's a nasty disease. So these are the mice section through here of the mice that got the control, the carrier molecule. And I know you're not all pathologists, but what you're seeing here is a big thickening of the muscle wall. You've lost all architecture of the villus in the intestine. And all of this is T cells coming in that are attacking your own tissues, causing this ulcer. In the mice that got the sugar, you can see that the cell wall is not thickened. Here's your villus. So you've got villus architecture maintained, and there's no inflammation. Okay, so are these sugars then a common link in immunosuppression? We think they are. Again, found on these anti-inflammatory helminth parasites, found on tumors and cancers, and we have now 2 papers out showing that tumors and cancers express these same sugars and drive the same suppressive mechanisms. Found in breast milk and urine of pregnant women, and on viruses, including HIV-1. So we think this is going to be a new paradigm for the way that certain molecules have been shared throughout evolution between our own cells, cancer, embryonic, and pathogens, that drive immunosuppressive type responses. And the parasites and the pathogens, of course, have all used them to their advantage to shut down your ability to kill them. So let's go. Okay, so to summarize, the sugars found on all these things that I've just told you about. All cases are associated with induction of immune energy or a switch from type 1 to type 2 responses. Again, found in human milk, which is immunosuppressive, urine of pregnant women. And we hypothesize that expression of this Lewis X is a universal anti-inflammatory, found on autologous cells as well as pathogens. And the people that did the work, Paul Thomas and Michelle Carter, did all the dendritic cell work. Paul's out at St. Jude's on a postdoc. Olga and Luis did all of the suppressor work, and Akram helped me out with everything. And of course, we were funded by the NIH and NEOS Technologies. So I'll stop there as I've worn out my welcome. Yep, it's Charles Janeway, the 5th edition. It's a fantastic book, and what happens if you buy the book, in the back you get a CD, and the CD has all the figures from the book in it, and they even have them organized in PowerPoint presentations if you want them. So for me, with the classes I teach, it's a great teaching aid. They even have some movies in it. So the figures I showed you on antigen presenting cell slide, they actually have that as a movie, so you could use that for a teaching device. Charles Janeway, 5th edition, immunobiology. Yes?
One of your very first slides, you were talking about the natural flora on our skin, and I was curious about your comments about all the antibacterial products that are on the market.
Mm-hmm.
What is that doing anything?
Well, probably women are much more sensitive to this than men are, okay? But as you know, if you take certain things or certain medications, you alter the bacterial flora of the vaginal, okay? And what does that make you susceptible to? Yeast, okay? So that's a very good example of what happens when you alter the normal bacterial flora of an environment. Another opportunistic pathogen, which normally couldn't be there because you've got your regular bacteria, now could come in and invade that niche.
Is that happening? Do you see any evidence of that?
They see it with people. They can see it in people that do this because when they go in and they get a wound, they don't respond properly to normal antibiotics because they've already wiped out the normal flora that you would treat with an antibiotic. So they actually have newer type pathogens. So they have to actually go in and try to figure out what the pathogen is and then get the antibiotic for it. So a group has approached us about this, and lupus for me, I'm not an expert on lupus, but lupus, as I understand it, doesn't fit the Th1, Th2 paradigm completely. However, one guy said that he thinks it'll work. So there is a group at Harvard Medical School that works on lupus, and we've got the sugars, so we can just give them the sugars and try it. Yes? You know, we've done 3 diseases. We've also done diabetes, and we didn't show that here, but it works beautifully in diabetes. And we don't know the mechanism yet. In fact, that's what we're really working on now. But there's a whole other facet I didn't show you here called suppressor macrophages. It's a population of cells that come up in cancers, and the sugar drives expansion of suppressor macrophages. And we think that they might be what's causing you to down-regulate your T cells so that they can't attack your tissues. And it looks like it's a systemic response, so that when you give the sugar, you shut down these auto-self T cells all over your body, and you get better. So like anything that remember I told you about earlier, these anti-inflammatory treatments? I think the same would apply here. You'd have to figure out how often could you give them to stay in remission without making yourself so immunosuppressed that you then would be susceptible to other diseases. Yes?
A little connection with the I've read this so many times, I'm a little confused. So
Yeah.
why does that help?
Well, there's two schools.
Theoretically, an autoimmune disease?
There's 2 schools of thought. There's the hygiene hypothesis goes both ways. One is that you don't get enough bacterial exposure in life, therefore you don't drive enough pro-inflammatory responses. And there is evidence to support that, particularly by the Japanese. The other group says you're not getting worms anymore. Okay, and there's several lines that show that that's the case. Many of you are old enough to remember pinworms. Okay, so pinworms were ubiquitous in this country until about the 1980s, at which point they were virtually eradicated from most areas except for the southeast. No one has yet done an epidemiological study, but I would like to see, in fact, is there more autoimmune diseases in people that live outside of the southeast versus those that are there, because they have another worm that they get as well.
So if you're putting out, let's just say pinworms in the dirt, that means your body
Is getting your immune system getting educated in a way that it used to be getting as primitive people, and you've evolved with these parasites. You got to remember, humans evolved with worms, right?
So that means your Th2
Two-
responses are
Being accelerated and augmented. And for those of you that are dog owners, you're probably aware that autoimmune diseases in dogs have gone through the roof, okay? And what's happened with dogs? They're wormed, they're wormed, they're wormed, okay? So again, it's anecdotal, but when you talk to your vets about the increase in autoimmune diseases in dogs, it's gone through the roof as well. Yes?
When you treat the people with sugars, is that orally, intravenously?
We were doing this by intramuscular injection. We also did it in mice by peritoneal cavity. Of course, the drug company would want to go topical for psoriasis, and they'd like to be going oral because that's the least invasive way to do it. We do know that the sugars can go in through the oral cavity, and they actually pass out in the feces and they're intact, so that would work. We just haven't done it.
What would you suppose is this going to, if this all works out the way somebody with stock in that company hopes, is this something you take a pill a day, pill a week? Or?
You know, I'm a scientist, right? Yeah, I'm a scientist, and I'm not. But they did do a marketing study by a marketing group, and the way they see this is that they would take that real potent drug, that anti-TNF alpha receptor, that knocks everything down, and everybody goes into remission. But you can't keep giving it because you're going to get really ill, and that after you go into remission, they would then put you on the sugar to keep you in remission. Okay, so you know that's their marketing analysis, and again, that's way beyond my scope of where to put things. Okay. All right. Yes. Actually, we started out on vaccine development, and we have a large grant in that we hope we're going to do well on. That's to actually have a veterinary vaccine for schistosomiasis in China. It's already been tested in China. The disease is a zoonosis, which means it goes between large animals and man. And in fact, water buffalo account for 80% of the transmission of the disease. As you can imagine, one water buffalo dropping compared to one human dropping. With our collaborators there that we've been working with, they've tested our vaccines in pigs, sheep, and water buffalo. They're effective. So this grant would be to treat humans with drugs, treat the water buffalo with drugs, but then give them the vaccine with the idea of reducing transmission to a near eradication level. They do because of the Three Gorges Dam that's going to increase prevalence of schistosomiasis by about 1,000-fold. It's now, the Chinese government just came out and ranked schisto as the number 3 disease behind HIV and TB in China. And you know, they usually don't like to broadcast things like that, so it's going to be a big problem there. Yes? Okay, could you speak up a little bit? There's been several studies published in China showing that there's an increase in hepatomas. It's hard to dissect the two because, as you know, Hep C does lead to hepatoma on its own. And I'm not so sure that those studies are as rigorously done statistically as I'd like to see. The stronger link is in Egypt with bladder cancer. And there's a very strong link between schistosome infection and increased bladder cancer in Egypt. Okay, all right.